Disease, again, in the latitudes where the Anopheles mosquitoes are commonly found. Tropical latitudes down to the subtropical latitudes. So through Central and South America, Africa, Asia, prevalence of disease, again, associated with economic factors. There are, again, non-government public health efforts. A variety of organizations have taken up the challenge of providing mosquito netting for individuals in these endemic areas. Something as simple as putting netting around beds to prevent mosquito bites helps control the prevalence of malaria in these endemic areas. Several different species of the protozoa in the genus Plasmodium can cause disease, and these different species tend to have a geographic distribution, so knowing travel history of individuals that have traveled to an endemic area, or knowing the history of an individual as to where they live can help with the diagnosis in terms of the speciation of the organism. We can also identify the particular species based on characteristics in the infected red blood cells, their appearance in those. There is a life cycle of this protozoa that takes place. When the mosquito has a blood meal, it acquires an immature form of the organism called the sporozoite. And when it bites a human, it transmits that form to the human. This form will not survive in the red blood cells. This migrates to the liver, where it undergoes development in what's called the exo outside of the erythrocytes, the exoerythrocytic stage. So the developmental stage of Plasmodium is in the liver. So after the initial acquisition and mosquito bite, within the first couple weeks, this is where the protozoa resides. It goes through a period of maturation, and then more mature forms called merozoites, and don't worry about the terminology, just understand that this is a maturation cycle, that this more mature form infects the red blood cells, the erythrocytes. So this is the erythrocytic stage of disease, and this is the stage of disease that in most cases is symptomatic. When the fever, night sweats develop associated with the disease. And because of the replication of the organism in the red blood cells, the presentation of those symptoms, fever spikes, tend to be predictable. Once the infection in the erythrocytes has become established, the reproduction time of the organism, of the protozoan, is what causes then the spikes in the fever. Because as the organism matures in the red blood cells and then breaks out of those red blood cells, at those burst points, that's what causes the fever spike. And that burst point is then characteristic both of the degree of infection and the particular species. So it varies from individual to individual. And typically what we'll do when we're going to collect a sample from blood from a patient that has suspected malaria is you go back and look at their chart and look at their temperature profile. You can see that they have fever spikes every 16 hours. Well, after the next fever spike at 15 hours, you're going to collect your next blood sample. Why are we doing it 15 hours?
Just before the spike.
Because that's when the highest level of parasitemia of the plasmodium in the red blood cell is going to be present. So you want to optimize your chances of identifying the organism in the infected red blood cell. Blood's collected, you make a smear on a microscope slide, a couple different ways you can do that. You stain it with the typical stain that we use for differentiating blood cells called the Wright stain. And then you look at it under the microscope and you look for the various forms that are present in those red blood cells. There are, depending on the organism and the stage of development, you'll see what are called ring forms. This is the nucleus associated with the protozoan here. One of the species, Plasmodium falciparum, causes these banana-shaped erythrocytes during its infection. That's diagnostic of this organism. So you can identify to species based on what the infected red blood cell looks like. This is just another schematic that shows basically the same thing, that after the mosquito bite, migration to the liver, maturation of the liver, and then spread through the circulatory system where the red blood cells are infected. Treatment, depending on the different stages of the organism in the body, the points of maturation. Some of the drugs are only effective in the red blood cells. Individuals that have a significant effect on the liver and one of the organisms that cause the banana-shaped red blood cells, that Plasmodium falciparum, tends to have much more severe effect on the liver. So we would use different drugs to treat infection with that organism. Because of that life cycle, the exoerythrocytic stage in the liver, and then the erythrocytic stage, individuals that travel to an endemic area are given a drug like chloroquine, a quinine drug, to take to inhibit the organism. But it only inhibits the organism in the red blood cells. Individuals that travel to an endemic area are instructed to continue to take that drug for 3 weeks after leaving the endemic area. Well, why 3 weeks after the endemic area?
Incubation time is probably...
Because the first couple weeks it's going to be in the liver and not in the red blood cells. And if you stop taking the drug, then you can develop full-blown malaria because it develops in the liver, affects the red blood cells, and you develop typical malaria. Most cases that we see in the United States are in that scenario. Folks stop taking their prophylactic drugs too soon and develop malaria as a result of that.
Is there like any drug that will kill it before it gets to the liver? Or anything like that?
These other drugs have activity in the exoerythrocytic stage, but it is both of these types of drugs are most effective in the red blood cell stage. Questions about malaria? One of the things I didn't mention, the reason I, on my first slide here, had this, we talked about, someplace there that talked about 1,500 cases in the United States a year. So most of those were as a result of travel. There are a few cases that develop in individuals that have not had any travel to an endemic area. What has happened is the individual that becomes infected hasn't traveled, but the vector that infects them has traveled. What's the vector for malaria? Arnopheles mosquito.
Got on the plane? Really?
Booked himself a first-class ticket, and
Of course, got to first
off they
class.
went.
Wow.
In cargo containers that are shipped through air cargo. Those cargo containers are opened up. They contain fruit, vegetables, and mosquitoes survive. And then in the cargo areas of air cargo areas of the airports, those mosquitoes then can find a susceptible host. They contain the plasmodium because they've bitten somebody wherever they came from initially, and then that individual unfortunately will develop malaria as a result of acquisition from air cargo transmission of the mosquitoes. And again, it's taken some pretty good epidemiologic investigations to identify that sort of scenario. So these individuals that acquire disease without travel typically live in close proximity to large airports where there's international air cargo.
So those airline workers who are just dealing with the cargo, do they have to get immunized like a travel would before they go someplace? It seems like they're at the same amount of risk. And do the doctors notice this stuff right off the bat?
It is relatively uncommon that this happens, so the cargo handlers typically aren't on medication to prevent malaria, but they're educated to recognize what the symptoms are because they are at potential risk. So in that education piece, then they can help to their medical providers with their medical history that indicates that they're at risk for that.
Thank you.
Same vector, different group of microbe. Mosquitoes, different genera of mosquitoes, transmit the West Nile virus, a virus that was first recognized in the endemic area. This isn't a trick question. This isn't like Rocky Mountain spotted fever. Okay. The Nile Delta in Egypt is where the disease was first recognized. Transmitted by mosquitoes. This just shows a geographic distribution in 2006 when most of the cases of West Nile were in the central to the just west of the Rocky Mountains in the U.S. Relatively little, no disease in the east. Because these folks have already been bitten by mosquitoes and they are now immune, so that the disease has moved to susceptible populations from the East Coast. In 1999, the first cases of West Nile virus in North America were identified in the New York, New Jersey area, probably resulting from the same importation mechanism that we just talked about with malaria for the United States, probably an air cargo. Unlike malaria, though, there is another reservoir, another animal reservoir for West Nile virus. Most of you have heard of West Nile virus? Is there another animal that you associate that with?
Just a mosquito.
Typically in the spring, you'll start to hear notifications from health departments that ask for individuals that find dead birds to notify the health department, because birds are the other reservoir. Dead crows are typically the sentinel animal in terms of recognizing West Nile virus activity. So it was transmitted initially probably